it might take like 9 minutes and 58 seconds to do. So the way I could have done it.
Shall I keep this one for you, or do you want to sit?
Okay. Well, welcome back, everybody. Hope you had a cup of tea, biscuit, and welcome to our afternoon session. And this is the same as we had the last couple of meetings, which is an update on the MRC-funded research projects that have now been going on for about 3 years, if I remember correctly. And so we have our first of our speakers, and our first speaker is Nadia from Newcastle, and she's going to give us an update on Sjogren Syndrome and the like, fatigue in. Nadia.
Good afternoon, everyone. Can you hear me at the back? My name is Nadia Howard-Tripp. I'm an academic clinical fellow in rheumatology at Newcastle University. I'm giving this talk on behalf of Professor Fai-Ing, and I'm going to talk about the biological fingerprints of fatigue in primary Sjogren syndrome. I'm going to outline what this study is, a little bit about primary Sjogren syndrome, and update you on our progress so far. And chiefly, we're talking about genes and fatigue and Sjogren's today. So this study functions on the belief that there are common biological pathways that underpin fatigue, and that these pathways can be found in peripheral blood. And these pathways, the emphasis is on the immunological pathways. The aim of this project is to compare the biological profiles of patients with primary Sjogren syndrome who have varying levels of fatigue, to see if there are possible differences between those who experience high fatigue and low fatigue. And then the second stage would be to assess the potential utility of the biological fingerprints that we find in chronic fatigue syndrome, ME. So Sjogren syndrome is a chronic autoimmune inflammatory condition. It is a systemic condition, but the main features are oral and ocular dryness and profound fatigue. And in fact, 70% of patients with Sjogren syndrome report disabling fatigue. In addition to this, there is considerable symptomatic overlap with chronic fatigue syndrome. And in fact, some studies suggesting that up to 2/3 of patients with Sjogren syndrome actually fulfill criteria for chronic fatigue syndrome. As I've said, the burden of fatigue is huge in Sjogren syndrome, with the majority of patients ranking fatigue as being one of the most important symptoms which needs improvement. So this is an outline of our study. We have the UK Primary Sjogren Syndrome Registry, and this is a large patient registry which contains biobank serum samples, as well as a wealth of clinical and biological information. And we looked at patients with high fatigue and low fatigue, and this was defined by the fatigue visual analog scale, and high fatigue was defined as above 75 and low fatigue below 25. And we also looked at whole blood RNA and microarray data in order to perform the analyses in the box here, sorry. So the first thing we looked at was differential expression of genes, and it functions on the belief that, well, the fact that all cells contain the same complete genome, but only a small percentage of this is expressed in each cell. And we were wondering, is there a sort of differential expression of certain genes between those patients who have high fatigue and low fatigue? And this is what we found, and I'll talk you through each of these slides. So at the top here, an A, that this is a volcano plot looking at the different gene expression between high fatigue and low fatigue. And essentially this volcano is quite sort of inactive, and showing that there's actually no difference between the high fatigue and the low fatigue groups within this group, and it's also worth pointing out there were 38 patients in the high fatigue group, 21 in the low fatigue group, and we couldn't find a difference there. There were, however, differences in the next one in B between the Sjogren Syndrome patients and controls, as shown by the sort of eruption on the sort of top right-hand side of this plot, showing that there were differences in expression. In fact, 334 genes were differentially expressed in the Sjogren's group. The next plot essentially shows the mean sort of gene expression between the high and low fatigue groups, and essentially they all fall on top of each other in a line as it goes up, showing that there was no difference there. And again, in D, principal component analysis, we didn't find any significant difference between the high and low fatigue groups in terms of differential gene expression. The next thing we looked at was the type 1 interferon gene signature, which is being implicated in a number of immunological processes and diseases. It's also present in the majority of patients who have Sjogren syndrome. And interferon alpha treatment can induce it. So I think it's an interesting thing to look at here. What we found, however, with Sjogren syndrome was that there were no significant differences in the fatigue level based on whether interferon gene signature was active or inactive. However, there were slight differences between disease activity, the S-star, that's the Sjogren syndrome disease activity index there. The next thing we looked at was gene set enrichment analysis, which is essentially a sort of a statistical approach which identifies clusters of genes which may be functionally relevant to a particular disease. And again, we use these low and high fatigue groups. And the idea behind this was to try and identify some sort of fatigue-related metabolic pathway, and then after this perform a leading-edge analysis, which essentially is a sort of software package which picks out the core genes which could account for the different signals you see in the different groups. And we did actually find some results with this third analysis. So we found that actually there were 19 enriched pathways between the high and the low fatigue groups, and they're listed there with a p-value and a q-value, which refers to a false discovery rate. And this is a little bit more about these pathways. So there were 3 actin-related bioparticle pathways, 15 G protein signaling reactome pathways, and 1 incretin-related reactome pathway. And again, this may not mean that much to you, and indeed it does not mean that much to me either. But what we look at is behind these genes, what genes are responsible for these pathways, and that's the interesting thing that we wanted to look at, because a lot of these pathways are implicated in many different processes. And so this is a list of the genes that are implicated in the first set of pathways. And again, the leading edge analysis sort of identified the core genes that are in bold that we think are the most responsible for these different signals. And again, the purpose of this, and I know you won't be able to read it sort of all in the time of this presentation, and we'll come back to it at the end, but it is just to show you that these are the genes that we think are responsible for the differences we're seeing between the low and the high fatigue groups in this experiment in Sjogren syndrome, and that looking at these particular genes, or at least having identified them, we can perhaps focus on them and look at whether they exist in the existing literature. And if we go on, this is the next one. Again, the most important ones identified in bold here. And you can see this one here. So in terms of what all this means, so in the existing literature, several of these genes have already been sort of at least implicated in fatigue, and we feel therefore warrant further investigation in terms of their potential role in fatigue. Finally, to sort of confirm that these genes were important and were sort of the correct ones, we used a machine learning approach to try and look at these 55 genes, or collectively 55, that were associated with fatigue, and to see if we could sort of classify fatigue level based on the genes. And what we found from this is actually when we used all of the different genes behind all of the pathways, we didn't find that good an area under the curve, essentially. But when we picked out the leading edge genes, which are the 55 sort of important genes, we actually got a mean area under the curve of 0.866. And with these ROC curves, essentially you want to be as close to the top left-hand corner as possible, in order to sort of say that you're fairly confident that this sort of mechanism can actually reliably sort out your groups based on these genes. And again, we repeated this using just 50 random genes picked from the 181, and we got a similar area under the curve of 0.5, indicating that it is actually these 55 that are sort of accounting for these differences and able to sort between the fatigue groups. So this is quite interesting here, and this will be published in a recent paper by our group. So in conclusion, our standard microarray analysis didn't identify any particular signature of fatigue that we could find in terms of differential gene expression. However, as I've said, the sample size was fairly small with this, and it would sort of warrant repeat in another cohort. Gene set enrichment analysis, however, did implicate several potential fatigue-associated pathways and genes, and this was significant in this case. And our main conclusion is that further studies of these pathways, which I've mentioned, is required. This is a sort of outline of the Sjogren's project. So I've spoken today about this whole genome expression analysis. The other arm of the project is actually on serum cytokines and fatigue levels, and I'm actually going to speak about this tomorrow, and we've got some quite exciting data from this, and I look forward to sharing that with you tomorrow. Otherwise, I'd just like to thank everyone in the MRG. As I said, this is on behalf of PHI and everyone involved at the UKPSSR, patients, volunteers, our sponsors, in particular the MRC. I'd also like to thank you for listening, and I'm happy to take any questions.
Thank you very much, Nadia. Time for a couple of questions. Burning questions for Nadia. Over to you, Bernie.
So, hello there. You showed a number of genes of interest that were differentially expressed between the high and the low fatigue group. Do you have any favourite genes or favourite pathways that you'd like to investigate first?
Not as yet is the answer to that. I know that there has been a lot of work on looking at leptin and fatigue, and one of those pathways was leptin, and I know that one of my colleagues actually has a poster particularly looking at that, so that is an interesting place to start, but I at the moment, not that we don't have a sort of a top one to start with that I'm thinking of. Thank you.
Behind you, Sonia.
Nadia, sorry, I was not paying attention on how do you divide the low and high fatigue? Can you
So it's based on the fatigue visual analog scale, so the VAS scale, and that's 0 to 100. High fatigue was defined as a fatigue VAS of over 75, and low fatigue of below 25. The average level, the mean fatigue in this group was 55 in the Sjogren's group. So we were really looking at the extremes to try and definitely sort of separate them out. We did also look at two other measures of fatigue. So we looked at the fatigue scale on the ESPRI, which is a sort of Sjogren's measure of patient-reported index, and that's a Likert scale of 0 to 10, and also at the ProFab physical fatigue questionnaire. And we also ran continuous analyses looking at fatigue and looking at the differential gene expression with all those three measures of fatigue, and we didn't find any differences with them, which was why we went with the sort of the cut-offs for that. We did look at all the different other measures as well, but we felt that was that's right. Thanks.
Thank you, Nadia. I'm going to cut you off there. While Julia just gets plugged in, let me just give you food for thought how we think that actin binding proteins expressed in peripheral blood cells lead to fatigue, which, as I understand it, is actually a concept that lives in your brain. So I leave you to think about how we're going to discuss that later. So our next speaker is Julia Newton, who many of you will know, and she's from Newcastle, of course, here. And thank you, Julia.
Thank you. It's a pleasure to be here today. It's a little disconcerting to be here doing an MRC update when the MRC funding that came out of the original call, actually finished 2 years ago now for this project. So what I'm actually going to do is talk you through some of the ongoing outputs that have been from the project, and just to update people on the update that I gave last year. Oops. That's not mine.
Just pop through. That was in case people are asking questions on the method.
Oh, I see.
Yeah.
Oh.
Yeah,
You're going backwards, that's why. I think I've seen, if you go down.
That's right. Oh, technical. Sorry. To start off with, I just wanted to remind people, you can't actually probably see it from the back of the room, but the MRC funding that we were lucky enough to acquire about 4 years ago now helped us by funding a team of people to facilitate a project that carried out a wide range of investigations in a large cohort of over 50 patients with CFSME. That included 7 visits to the clinical research facility and included 3 MRI scans of brain, heart and liver. It also involved a number of nucleotide investigations, including plasma volume, red cell volume and MIBG. We also performed a full gamut of autonomic tests, a dexamethasone suppression test as a measurement of HPA axis function, and a full battery of neuropsychometric assessments, all in the same participants. So the considerable advantages of that mean that we have the ability to look at a whole systems approach to CFSME. The challenges that has given us, considering it was 2 years funding and we used the full 2 years to recruit the participants, is that the analysis has taken us a huge amount of time because that's been sort of done on a wing and a prayer. So my co-investigators, I'm hugely grateful to because they've really facilitated a lot of where we are today. So Andy Blameyer is the MR physicist, Stuart Watson is senior lecturer in psychiatry, and Peter Gallagher is a neuropsychologist. And Andreas, who is still working in this field subsequent to this project, is here with us today, and he's got a talk on the brain MR results tomorrow. Laura McLaughlin, who was the CRA who led the project, is now working as a postdoc in Sweden, still in the field of CFSME, and she completed an MD as part of this project. So the MRC funding that we were lucky enough to receive facilitated and underpinned a huge programme of work that goes on today. And I just wanted to give you a sense of what sorts of things have come off the back of the MRC funding. So Nadia's talked about the Identifying Biological Fingerprints of Fatigue project, and that's gone on to allow us to use those samples to facilitate research in a range of different other projects in Newcastle and beyond. ME Research UK have funded a project with Faisal Khan in Dundee, and we're using samples to help him. Action for ME have funded Joe's project, and she's going to talk about that tomorrow. ME Association, using some of the same samples and additional samples, have also funded a project that was initially carried out by Sarah Jane Bolton and has allowed us to form a close collaboration with Carl Morton and his group in Oxford. And the actual MRC cohort that we developed has gone on to receive funding from the MRC Confidence and Concept Scheme, and we're in the process of utilising the cardiac data at the moment as the basis of a DPFS application, which we hope to submit in the new year. We've also combined all of that with money from Action for ME, where Cara, who's here, and her predecessor Gina, who were PhD students, have been developing projects looking at muscle function, and that has also received confidence in concept funding for Audrey Brown, who was the lead author on our muscle paper in PLOS ONE. And I was delighted to hear on Friday from the MRC that the work from that has now been invited to full application, which we're submitting in the March round. Action for ME also supported work that was a PhD student, Zoe Gott, who again is here, and that went on to be an ME Association funded intervention study, which has recently been published. And then the final piece in the jigsaw for us is the concept that the MRC project couldn't necessarily be generalisable to the wider ME population. So ME Research UK funded a project to look at which components of the MRC protocol could we take out to homes and perform assessments in severely affected housebound and bedbound ME patients. And Vicky Strassheim, who has done a workshop just before this and is talking tomorrow, will illustrate some of the complexities and difficulties that we've had delivering that project. So these were the main objectives of the project, which set out to look at the pathogenesis of autonomic dysfunction and chronic fatigue syndrome and the relationship between autonomic dysfunction and cognitive impairment in CFS. I'm just going to go through each of those objectives to let you know what the data is leading to. We're now working, well, Andreas is working on 3 papers looking at the brain MR findings. I have to say the vast majority of the findings are negative in terms of brain MR. The HPA axis function has had a range of different students working on that as MRES projects, etc. And Cara has done some work, and the data from that again has been negative. The cardiac studies, the cardiac MR and the MIBG studies have been very importantly positive, and I'll talk about those in a second. And similarly with the plasma volume and red cell mass studies. The other additional thing that we've done is develop a number of new methodologies around liver MRI and brain MR, which again will allow us to publish methodological papers in the fullness of time. So this just summarises the assessments that patients underwent as part of our protocol to, I hope, highlight to you the fact that we've utilised every single bit of the input that the participants so graciously gave to us, because it really was quite hard for people to participate in this study. And I think without the dedication of Laura and the rest of the team, it would have been really difficult for us to get to the point where we've got to. And one of the big things that we learned from this project is that doing research with this population is really doable, and that people really want to participate. For those of you that are used to doing clinical research studies, a recruitment graph like this is usually unheard of. There's usually a divergence of those 2 lines of what your target is and what your actual recruitment is. And this is something that I think is testament to the fact that the team formed really good relationships with the local patient support groups and the patients themselves, so that 100% of the participants completed all of the assessments. And this is the latest publication. This was published in Open Heart in June, and since then we've had 10,000 downloads of this paper. So to put that into context, that's the same number of samples we're wanting to recruit as part of the mega collaboration. So 10,000 people are interested enough in the hearts of patients with CFS-ME to actually download this from the website. And the conclusions of this study were that patients with CFS-ME have small hearts, small cardiac volumes, something that we've shown before and others have shown with other methodologies. That people with ME have low plasma volumes, and that the lower your plasma volume is, the more fatigued you are. And this is the basis at the moment of a study where we're looking at expanding plasma volume with oral fluid and intravenous fluid, which is funded by Action for ME, and as a proof of concept, in the hope that that will underpin the DPFS that we're planning to submit in the new year. So my last slide, I'm not sure whether I should keep being invited back to give updates when we're 2 years beyond, but I'm always delighted to do that. I think there's a huge amount that has come out of the MRC funding that we were lucky enough to receive. Those outputs are tangible in terms of papers and grants, which is the currency that in academia we're judged by. But there are other things that it has led to, which are also very important. It's brought us credibility in Newcastle, and we now have a Newcastle University Fatigue Research Centre, which is a recognised research centre. It's cross-faculty, and that gives us a small amount of funding to allow people to come to meetings and to facilitate fellows and researchers to come and work with us. It's facilitated a huge number of national and international collaborations. It's great talking to people about the science we've done and helping facilitate their science. So we have strong collaborations with colleagues in Poland, Australia, the US, and that can only be good for the field, and I think that has come directly on the back of the opportunity that the funding gave us. So as always, I just want to finish off by saying thank you to the funders. Without the support of the charities and MRC, we wouldn't be in the position that we are at the minute. You know, on Friday when I received the email from the MRC, having had so many, sorry, no, we're not going to invite you through to full application emails, to actually open the email at 4 o'clock on a Friday and see you're invited through to full application, it makes it all worthwhile. So thank you, and I'm happy to answer any questions.
So,
Julia, you in your brief summary of HPA, you said that you got negative
Yes.
findings. Does that mean that what we can't regard as perhaps one of the most consistent findings of HPA dysfunction, you found different results?
So we've now looked at HPA axis function in 2 different ways, and with the dexamethasone suppression test, we've looked at not only cortisol levels but a range of different cytokines to look at whether or not there are changes when we depress HPA function, and we have not been able to find any changes that are significant in CFSME compared to controls. We've also taken a cohort of patients where we've got salivary cortisols over 24 hours and looked at circadian rhythms, and again not been able to find abnormalities in CFS versus controls. Cara is at the moment writing that up, and will hopefully, it's always very difficult to get negative studies published,
Yes.
which is unfortunate, but we will try.
Yeah. Is there a simple reason why you're different to everyone else?
Well, I think, the main reason we're different is that our cohort screened out rigorously for depression. So, sorry, I should have made that clear, is that the cohort, the 50 participants, all had a SCIDS, so we know that they don't have any history of depressive illness, current or past. So it may be that the presence of depression as a co-founder is the complicating factor.
thank you, Jim. We're going to move on because we have 4 more presentations to come, and then time for questions after. So if you have questions, can you hold them, because the speakers will remain there waiting for your questions. So next up is Alice Russell from King's College, and Carmine is enjoying himself in Italy yet again, and Alice is here to represent the group.
Yes, I'm covering again, I'm afraid. Last time he was ill, this time he is working, a bit in Milan. But he sends his apologies, and it's really nice to be here to share a little update with you. And unlike, oh, that's not mine, that's very lovely. It's not Camberwell though, where
That's
I'm
Newcastle.
based. That's Newcastle, it's definitely not South London. So unlike what Julia was saying before, I think we were a little late starting in this one of MRC studies, and we had an extension, so we just finished our last bit of data collection last month. So I'm just going to share a little bit of the preliminary stuff with you and just a bit of background about the model as well as a refresher. So we're looking at persistent fatigue induced by interferon alpha as a new immunological model for chronic fatigue syndrome. And I don't need to tell you about the link between CFSME and immune dysregulation, so I'll skip that so I can share more results. And we talked a lot today about the problem, as it were, one of the problems in CFSME research, where we've learnt a lot from cross-sectional studies, but what we really need is prospective cohort studies. And we've also talked a lot about the ideal kind of prospective cohort studies, where we can follow people and see who becomes ill with CFSME and who doesn't, and what are the factors involved. But we're proposing a slightly different kind of prospective study. And like we've heard from the other speakers as well, it's about looking to other conditions to see what we can learn from them to better inform our study of CFS-ME. So what do we think the solution is? In our case, we're looking at interferon alpha, which is a pro-inflammatory cytokine. So it's part of our natural immune response, but it's created synthetically and then used therapeutically in the treatment of hepatitis C and some cancers as well, but it's Hep C that we're interested in. It was until recently, and kind of still is, the standard treatment for hepatitis C, and the patients have to inject it every week for 24 to 48 weeks, so 6 months to 12 months, usually 6, and it boosts the natural immune response and inhibits replication of the virus, but not without a wide range of side effects, and this is what we are interested in. Most patients do recover post-treatment, so the treatment stops. The doctors tell them you'll be fine within a few weeks once the medication's out of your system, but it's not the case for everybody. So a survey conducted in 2010 by the Hepatitis C Trust found that about 60% of patients are still reporting problems with fatigue 6 to 12 months after the treatment has stopped. And about 40% of patients a year after the treatment stopped. And it's not just fatigue, it's a range of symptoms that are very similar to what we see in CFS-ME, and that's why we think this is an interesting proxy model, as we call it, to use for CFS-ME. So the aim of the study was to use interferon alpha-induced persistent fatigue as a proxy model to understand how an exogenous inflammatory trigger, so the inflammatory trigger here is the treatment, not the virus, leads to a pattern of biological and behavioural changes that is associated with what we're terming persistent fatigue. So obviously we're acknowledging it's not CFS-ME, it's persistent fatigue that we're interested in here, even when that stimulus is no longer present. So again, we've talked today about how a lot of patients might identify some kind of immune trigger as kind of the start of their problems, but we know they don't have it anymore, but they're still experiencing these symptoms. And then we're comparing these Hep C patients with a sample of CFS-ME patients and controls. So the first part is a prospective cohort study, as I said. We've followed a cohort of Hep C patients undergoing this treatment, recruited from one of 4 sites across London, and we've seen them a minimum of 6 times. So we see them before they start the treatment, immediately before their first injection, and then monthly during the first 3 months of treatment, and then at the end, and then we see them 6 months post-treatment as well. And we expect from the survey data about half of them to still be experiencing problems with fatigue and associated symptoms when we see them 6 months post-treatment. The second part is the case control cross-sectional study. So that's where the CFS-ME patients come in. We aim to get 50, either from the South London and Maudsley service in South London or the Royal Free Service in West London. And then aim to see the healthy controls as well, 50 of those, and that would be for each group a one-off visit where they did all of the measures that we did with the Hep C patients over time. Combining the recruitment with an earlier study and this most recent study, we saw we enrolled 108 hepatitis C patients, 18 of them we couldn't include, they stopped treatment too early, withdrew, were excluded because of health conditions that developed. So yeah, we had like 90 that we saw. I'm going to tell you about a subset of these patients in a moment. And then we had a final sample of 52 CFS-ME patients and 58 healthy volunteers as well. And this slide, it just shows you the pattern of fatigue over the course of interferon alpha treatment. So this is just the Hep C patients. And we've used the Childer fatigue scale to categorise the patients into those whose fatigue resolved. So when we measured their fatigue 6 months post-treatment, had it got better or at least returned to baseline levels, and those whose fatigue persisted. So these are defined as a group of patients whose fatigue was worse than baseline 6 months after that stimulus was no longer present. Just to give you a bit of background about socio-demographic characteristics, we didn't find any significant differences between our patients. Mean age of about 45 years, predominantly male, I know that's very different to CFS group, between 80 and 90% male, mainly white, so slightly higher proportion of white individuals in the persistently fatigued group. Sorry, I should have said this is a subsample of 55 of the patients. Educated to degree level, slightly more in the resolved fatigue group but not significant. Similar levels of unemployment at about 30 to 40%, and similar rates of patients who were married or living with their partner. We also looked at history of depression, family history of mental illness, and whether they had depression that was induced by the treatment. About 30% of people generally do from the treatment. It's another one of the side effects that's known. And again, no significant differences between the groups, although slightly higher rates in those who were persistently fatigued. No significant difference in the genotype of the virus. There are different genotypes of hepatitis C infection. We looked at the level of virus and the degree of damage to the liver via a FibroScan, so 2 markers of disease severity. We didn't find that one group was more disease than the other. The only trend was that those patients who had longer treatment, so longer exposure to the inflammatory trigger, were more likely to experience persistent side effects post-treatment. No difference in the treatment type. And then slightly more interesting, I think, for me anyway, there are 2 markers of clinical response in these patients under this treatment. The first is what we call rapid virological response, if I can say it. So had the treatment worked 4 weeks into the treatment, into this 6 months treatment. And again, not significant, but there were slightly more patients for whom the treatment worked quicker in the resolved fatigue group than the persistent fatigue group. And that's important, I think, because of what's starting to come out from the early analysis is perhaps this early response to the trigger might be the most important thing in this group. And interestingly for us, and contrary to what our consultants, our liver consultants expected, it's not that the patients still have the Hep C and that's why they're so tired. So actually, although it's not significant, if anything, the persistently fatigued patients were more likely to have cleared the virus. So they've not got Hep C, they're not under the treatment, but they're still feeling very poorly. So that's what we're interested to know, why is that? And I'm probably talking for too long, but I'm just going to show you some results that is also in my poster. 2 minutes. Also in my poster, if anyone wants to chat about what they've seen or have a look at that with me. To put it very simply, we talked about cytokines, protein messages of the immune system. They do a whole lot more than that, but just as a kind of basic overview, we measured them in serum using the mesoscale discovery or MSD V-plex assays in a group of 35 of our patients, so 24 of these patients whose fatigue resolved and 11 whose fatigue persisted. And we measured it at baseline, so pre-treatment, and after 1 month of treatment, 6 months of treatment, and then when we saw them 6 months post-treatment. And for these markers that I'm going to show you, so interleukin 10, there was an effect of interferon alpha treatment. It did change over time, and the levels were higher in the persistent fatigue patients. And if I could draw your attention to that change from baseline to week 4, so that first month of treatment, having a more exaggerated response, which we're very interested in. But come 6 months post-treatment, they're clinically reporting different symptoms, but they're not more inflamed. And similar patterns as well in interleukin 17A, higher in the persistent fatigue group, and IL-6 as well. Again, both experiencing a change, but the persistently fatigued patients are higher, but no different at the 6 month post-treatment point. IL-7, slightly different. It did go up after the end of treatment in that 6 month period, but not by a lot, but there was an effect of the time group there. So that's it for today. We hope to have some more results to share with you soon. But yeah, thanks very much to my colleagues and of course all of the patients who made it possible. As Julie was saying, if anything, my main problem as coordinator of this study was too much enthusiasm. So just trying to keep tabs of all of the people that really wanted to take part but were not able at that moment, and kind of trying to keep them involved, which was a wonderful position to be in. So yeah, thank you.
Alice, any burning question for Alice? Or we're going to race on because we're getting behind and we're going to have. Thank you. Thanks. So our next speaker is Anne McConnell, and she's from Liverpool, and she's going to give us an update of what you've been up to.
Thank you. Bottom button.
Thank you, and thank you for the opportunity to present our work to you here today. I'm based in the Institute of Ageing and Chronic Disease at the University of Liverpool, where we also have the MRC-funded ARUK Centre for Integrated Research into Musculoskeletal Ageing. Hopefully by the end of my very rapid talk, I'm going to have convinced you that we have some real similarities with what we're seeing in the development of muscle fatigue, muscle dysfunction in chronic fatigue compared with old age. Oops. I don't think I need to spend so much time on this slide. We all know that chronic fatigue syndrome is very poorly understood and is associated with this prolonged debilitating fatigue. And so, how do our muscles work, first of all? How do they generate force, and what happens when they go wrong? Just very briefly to show you the structure of our muscle. So if we take a long muscle, you can break that right down to an individual muscle cell or a muscle fibre, which runs almost the length of the muscle sometimes. So very large structures within our body. Muscle bulk is quite a large proportion of our body as well. We need to remember that when I start to talk about the sorts of things that muscle can produce and the effects of those. So if we break it down even further, you see this pattern of proteins within a muscle fibre, and we have this mainly two proteins that are overlapping. So we have actin and myosin overlapping. When you contract muscle, those proteins engage with each other, and that's what generates the force. So on this next slide here, I'm just going to show you that in a little bit more detail. So it should run, yeah, good, the video works. So we have myosin here engaging with the actin filament here in muscle. When it engages, it generates force. The things that you see flying in here, one of them is energy in the form of ATP, so you need that ATP to generate the force. That comes primarily from the mitochondria, which are very closely embedded in the muscle cells and right next to the contractile proteins. So mitochondria are very important in generating these contractile forces and producing energy. We all know that our muscles can adapt, and Arnie wasn't born like this. He's worked very hard to generate those sorts of muscles. So when you're young, when you're healthy, your muscles can adapt. I haven't got time to talk to you about how our muscles adapt and the general mechanisms by which they adapt. But suffice to say that free radical generation is particularly important in this adaptation and this maintenance of healthy muscle. This is primarily work by Professor Jackson at Liverpool. And what he's done is teased out all of the pathways that are responsible for production of free radicals within muscle. You might remember, or you might think of free radicals as bad molecules. Actually, they're really crucial for this adaptation that occurs in muscle and for healthy muscles. What he's done is identified that the site of generation of free radicals that is particularly important in producing this adaptation is this plasma membrane NADPH oxidoreductase. You might think it's mitochondria, but it's not in this situation. So what have we learned from old muscles that we can take to our chronic fatigue patients? Well, as I said, you can take a muscle, you can hypertrophy it, you can make it bigger. Our old muscles can't do this. And the reason they can't do this is because they just can't respond to these signals. They don't actually have the signals. So if we look at protein generation, changes in gene expression with contractions, this is just one of the chaperone proteins that's involved in this hypertrophy of muscle. In a young muscle, you see rapid increase in this protein. In old muscle, it just doesn't respond. It doesn't produce these proteins. And again, the main reason for that is because in young muscle, this is what you see. Again, I don't have time to tell you about this, but we can isolate individual fibers, look at free radical generation in real time. What you see is this increased free radical generation in young muscles. It's already elevated in old muscles. And that blocks that signal. We don't get an additional signal in old muscles. What you have is a chronic activation of NF-kappa B in these muscles. And they're actually churning out cytokines in our older muscles. Further evidence that free radicals are involved in this process in young muscles comes from treatment of individuals with vitamin C. So a very simple treatment. We can, before the treatment, we can activate their muscles to adapt and to hypertrophy. That's shown here. We see another increase in one of these chaperone proteins. If we give vitamin C for 6 weeks at an over-the-counter dose, we block that. So clear evidence that free radicals are involved in that adaptation. So where does this take us with our chronic fatigue studies? So just to give you a little bit of background of this study, we're also just finished our MRC-funded work, and so we're still working on the data, and we have a very skilled statistician in Leeds working on this data with us. So our criteria were relatively loose. We took an 18 to 55-year-old range, some exclusion criteria, but we wanted to capture a whole range of patients. These data come from my colleagues in Leeds, who are nutritional psychologists who are involved in the study, and they've classified our patients in terms of symptoms at diagnosis, and so we have information on various questionnaires that they undertook. Similarly, we have information on subjective fatigue, physical activity, anxiety, depression, and so on, and this is going to be particularly important when we tease out differences related to the work that the statistician is doing with us. When we biopsy muscle, we have a whole vast experience at Liverpool of various analyses of skeletal muscle with our colleagues at Liverpool, John Moores, looking at whole muscle function, both voluntary muscle contraction and electrically stimulated contraction, bypassing the nervous system, looking at force generation, through to muscle biopsies where we can look at the live generation of free radical species from isolated fibre bundles, right down to skin single fibres where we can bypass all of the energy needs for that muscle fibre and look at the active myosin interactions. We also, in a cohort of 100 patients and 100 healthy volunteers, looked at cytokine panels. The reason we did this is particularly important. So we know that muscle weakness and exercise intolerance is a secondary consequence to many inflammatory disorders. Cytokines such as TNF play a major role in modulating the function of muscle. And we now know that muscle can be quite a major endocrine organ, producing a vast array of cytokines. So what did we see in our cohort? I should say that the muscle function, muscle biopsy studies were done in a subgroup which was fully powered, a subgroup of 17 patients. Mitochondrial membrane potential was relatively normal in our patients. When we looked at mitochondrial hydrogen peroxide production, it was tantalizingly elevated in all respiration states of the mitochondria. This is pooled samples, male and female subjects, and so we are teasing out those data at the moment with this statistician. As has been previously reported, and as we see in old age, voluntary contraction, voluntary ability to generate force is reduced in our patients. When we electrically stimulate the muscle, bypassing the whole of the nervous system, it's not reduced, and that was a similar effect that we saw in the skin fibres as well, very similar to what we see in old age. These data are quite new, and again are pooled data, but we're starting to tease out those in terms of the subgroups. And we see again some tantalizing increases in the serum cytokine levels in our patient groups. I pulled these out separately just because of the scale bars on the side for some of the levels. So going back to our old age study, we've got this altered ROS generation, hydrogen peroxide generation by mitochondria associated with this chronic NF-kappa B activation. In old age, and this is in mice, we have that's associated with, and in fact causes an increase in a whole panel of cytokines within muscle itself. So if we take one of those cytokines, sorry, I am trying to rush through this to give you a taster of our data, but if you take one study where we've taken mice and we've treated them with TNF-alpha, ignore the HSP10 treatment, which actually was very successful. Treat them with TNF-alpha, the force generation is significantly down. So, oops. More evidence that the environment of the muscle is particularly important in generating force comes from a study where we transplanted muscles, young to old, old to young, changing that environment and changing that inflammatory environment of the muscle. The take-home message from this is that it's completely dependent on the environment. So you take a young muscle, put it in an old mouse, it behaves like an old muscle. And we think that that inflammatory environment's playing a major role there. So going back to mitochondria, if you treat muscle cells with TNF, the mitochondria generate more hydrogen peroxide. We've got a drug treatment at the moment where we can block that increase, that TNF-induced increase. If you look at the cytokine production by those muscle cells, treat them with TNF, we see, in most cases, significant elevation. If we block the mitochondrial production of hydrogen peroxide, in most cases, we block that production by muscle. So what happens in our patients? When we look at the message levels for cytokines in the muscle biopsies, these 4 of them, interesting that it picks out IP10 and IL-6 again, following Alice, but we see elevations, significant elevations in these cytokine levels in the muscles. So our future work is really focusing at the moment on identifying the subgroups of chronic fatigue patients with altered cytokine profiles. We're also wanting to try and understand more the local effects of changes in muscle production of cytokines. We have some supplements that we've already been developing for our old age studies that we could potentially apply to chronic fatigue. In old age, and this is just an image showing an individual muscle fibre, every muscle fibre is innervated. And we're focusing on what might be going wrong at this level in old age. And it might be that that environment is changed in our chronic fatigue patients as well. And this really just shows a normal innervation, a nice pretzel-shaped neuromuscular junctions. This is what old age looks like. Finally, really would like to take the opportunity to thank the MRC in particular and the ME Association for funding the chronic fatigue work and all of the other studies funded by these other groups and the research group that have carried out the work that I've spoken about.
Thank you very much, Anne. So please can, if you have questions for Anne and other speakers, please hold them, make note of them, because we're going to have time for questions, but I think we should probably hear all the talks first and then move on to the questions. So our next speaker is Sue Wilson, comes to us from Imperial College, and she's going to give an update on what she's been up to.
Okay, so we did a study of sleep, and we looked at slow wave sleep and daytime functioning in chronic fatigue syndrome. The project was delayed for various reasons, but we've now finished it and we've got the data. We've analysed some of it, and I'm going to try and tell you something about that today. So why did we do it? Well, sleep problems are very common in CFS. Everybody says that they have problems with sleeping, and often they feel that if they've had a better night, they might be better functioning during the day. There's some quite good evidence that the structure of sleep, in that, looking at drilling down into the microarchitecture of sleep, there may be some deficiencies in very slow waves in slow wave sleep in people with CFS-ME. We wanted to do a test where we pharmacologically, artificially enhanced this slow wave activity in slow wave sleep, and then just look and see whether it makes any difference to functioning during the day. It was a very, the data collection for the study was quite difficult. Our participants came in for 5 days and 4 nights, twice. On one occasion they had sodium oxybate, which is the drug we gave to increase slow wave sleep, and on the other they had placebo, randomised, double-blind design. We recorded their sleep overnight, every night, but on night 1 and night 4 it was a full recording with EEG so that we could look at these slow waves. The other nights they just had respiratory recording for safety. And then we tested them on the day after, on day 2 and day 5, to see whether there was any difference in their daytime function. And we did various things. We did a multiple sleep latency test, which looks at objective sleepiness by giving people an opportunity to nap 4 times during the day with their EEG electrodes on, and then measuring how long it took them actually to go off to sleep. We did some grip strength and grip fatigue. We did some cognitive testing using pencil and paper tasks. And you can't see the bottom of the slide, but we did do lots and lots of visual analog scores of mood, of exhaustion, of irritability, of sleep, lots and lots of different subjective scores. So the flow chart, 331 people got in touch with us. And then lots of them dropped out after we sent them the information. We got 29 in for screening. They had to be screened overnight with their EEG, so we knew that they didn't have a comorbid sleep disorder, which we could exclude. And 10 completed the study. So this is a very interesting part of the study, the screening outcomes. So a large number, oh sorry, what have I done?
I don't know. Do you want to keep talking?
A large number of our patients actually were excluded for sleep disordered breathing at the screening stage, much more than we would have expected. There were other sleep disorders like restless leg syndrome, which also we had said in the exclusion criteria we'll exclude these people because they have a comorbid sleep disorder which is going to confound things. And these figures were very high, and we had to screen quite a few people in order to get our final subjects. Now, what about subjectively their sleep was poor. They were saying the Pittsburgh Sleep Questionnaire score was over 5, which indicates poor sleep and various sleep disturbance. So that wasn't particularly different between the people we excluded and the people who came into the study. But they weren't sleepy in the daytime, unlike the reports from other groups. Our final participants on the right-hand side had quite low Epworth scores. And if you look on the other side, on the left-hand side, you look at the people we excluded. So the high scores had been already excluded by having other disorders. So what have we found so far? Well, we did manage to increase slow-wave sleep per hour quite significantly on both the first night and night 4. Incidentally, the very slow component of the sleep, we've started to do the spectrum analysis, and it looks like that was also increased too. But what we also seem to have done is to increase waking during the second half of the night significantly. What about daytime function? Well, we haven't analysed it all, but morning sleep satisfaction showed no difference between drug and placebo. There was no difference in objective sleepiness, so the mean sleep latency during the day wasn't different after the drug or after the placebo. We haven't found any difference in grip strength. We haven't looked at the fatigue scores yet, fatigue data. And we did the Trails test, pencil and paper test of executive function. There is no difference with that either. The computerised CANTAB tests haven't been analysed yet, so we haven't got the results of those to show you today. So our analysis plan, get that done, it will be done by Christmas time, and look at all the visual analogue scores, because we haven't started, they're all entered, but we haven't started looking at them yet. So far, we've just looked at the mean of the people who completed and compared between drug and placebo, but I think because of that finding of increased wakening after the drug, we may have to do a secondary analysis looking at all the daytime function measures in the light of all the sleep variables, and that's going to take a little while as well. But we also think that we need to tell people about this high comorbidity of sleep disorders among people with chronic fatigue syndrome. So we want to try and get people together and have a meeting and talk about how can we push this forward, and also what are the implications for treatment in terms of patients' well-being. We're grateful to Action Friendly very much so for helping us recruit, but all the other organisations too, and the staff in the clinical research facility, and our wonderful volunteers who did cooperate and really were very patient with us. Thank you.
very swift resume of our type of work.